Have you ever wondered what you're really made of? I mean, deep down at the most fundamental level. Well, today we're going to dive right in and decode the building blocks of life itself. No, really, think about it for a second. If you look past the skin and bones and all that, what are the tiny microscopic nuts and bolts that put it all together? The answer, it turns out, is way simpler and honestly way cooler than you might imagine. That's it. That's the secret. All of the unbelievable, staggering complexity of life on Earth, from a tiny little bacterium to a giant blue whale, it's all written using a simple alphabet of just 20 chemical letters. We call these letters amino acids, and they spell out the instructions for building the little machines that run our bodies, proteins. Okay, so before we can learn to read this language of life, we've got to understand the letters themselves. I want you to think of them like Lego bricks. Each one has a standard way to connect to any other brick, but each one also has its own unique shape and personality. So what does one of these letters actually look like? Well, every single one of the 20 has the exact same backbone. You've got a carbon atom in the middle, and it's holding on to two groups, an amino group on one side and a carboxyl group on the other. These are like the standard bumps and holes on a Lego brick. They're how they all connect. But the real magic, the part that makes each one different, is that R group. That's the special bit, and it gives each of the 20 amino acids its own unique chemical personality. And here's a really cool trick these molecules have up their sleeve. Inside your body, which is mostly water, that amino group actually has a positive charge, and the carboxyl group has a negative charge. It's like a tiny magnet with both a north and a south pole. We call this a Zwitter ion, and because those charges cancel each other out, the whole molecule is neutral. It can just float around in our cells perfectly balanced. It's just super clever how nature designed it. Okay, so 20 different R groups. How on earth do we keep them all straight? Well, it's actually pretty simple. We just sort them into 5 families based on their personalities. You know, how their R groups feel about water. So let's meet the families. First up, meet the greasy gang. These guys are the introverts of the amino acid world. Their R groups are nonpolar, which is basically a chemical way of saying they're oily or greasy. And what does oil do in water? Right, it avoids it and clumps together. That's exactly what these amino acids do. They're hydrophobic, water-faring, so they bury themselves in the middle of a protein, hiding from all the water in the cell. This is a huge deal for helping proteins fold up correctly. So who's in this gang? We've got glycine, alanine, valine, leucine, isoleucine, methionine, and proline. I know, it's a bit of a mouthful. So here's a little trick to remember them. Just picture this scene. Grandma always visits London in May for proline's party. Say that to yourself a couple of times, and boom, you've got the whole greasy gang memorized. Next up, we've got the aromatic trio. These 3 are pretty special because their R groups have these big, bulky ring structures. They're also mostly nonpolar, so they like to hang out with the greasy gang. But here's their superpower. Those rings are amazing at absorbing UV light. And this is so useful for scientists. They can shine a special light on a sample, and the more of these amino acids are present, the more light gets blocked. It's a quick and easy way to measure how much protein you have. How cool is that? Our trio here is phenylalanine, tyrosine, and tryptophan. Now, the memory trick for this one is a little creative, but stick with me. Just think about the smell, the aroma of fine pine and yellow timber. Okay, so fine starts with F for phenylalanine. Easy. Yellow starts with Y for tyrosine. And tryptophan? Well, it has the biggest kind of wide ring structure of the 3, so it gets the letter W. See? Not so bad. All right, let's move on to a much more sociable group, the polar pals. Unlike the greasy gang, these amino acids absolutely love water. Their R groups have atoms like oxygen in them, which lets them form these little connections called hydrogen bonds with water molecules. They're definitely polar, but they don't carry a full-on electrical charge. So the polar pals are serine, threonine, cysteine, asparagine, and glutamine. The memory trick here is a fun one. Santa's team crafts new quilts. But hey, pay special attention to that C in crafts for cysteine. This one is a superstar. 2 cysteines can link their R groups together to form a super strong bond called a disulfide bridge. It's like molecular superglue, locking parts of a protein together. And finally, we have our last 2 families, the ones that carry a full electrical charge. You've got the positive pals, lysine, arginine, and histidine, and the negative Nancy's, aspartate, and glutamate. These charges are absolutely vital for how proteins grab onto other molecules. For the positives, just picture a little drama. Lysine likes arguing with histidine. And for the negatives, just think of 2 things you might see on a food label, aspartame and glutamate. Okay, so
We've met our 20 unique Lego bricks. We know their families, we know their personalities. Now for the fun part. How do we actually start building with them? How do we link these letters together to spell out a word?
It all happens through this beautiful, simple reaction. You take the carboxyl end of one amino acid and line it up with the amino end of another. Then, you squeeze out one molecule of water. And, click, they snap together, forming a super strong connection called a peptide bond. And you just do that over and over again. Click, 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 building a long chain.
But here's the thing, and this is maybe the most important part of this whole story. A long floppy chain of amino acids, well, it's kind of useless. The real magic, the part where life actually happens, is when that chain folds up into a very specific, very intricate three-dimensional shape. It goes from being a string to being a machine.
This folding process happens in 4 stages. First, you have the primary structure. That's just the list of our letters in order, the basic recipe. Then bits of that chain start forming simple repeating shapes like little coils or zigzags. That's the secondary structure. After that, the whole thing folds up into one big final 3D shape. That's the tertiary structure. And sometimes a few of these finished chains team up to form a giant supermachine, and we call that the quaternary structure.
Let's just zoom in on that secondary structure for a second. The 2 most common shapes you'll see are the alpha helix, which is like a tight coil or a spring, and the beta sheet, which looks more like a paper fan that's been folded back and forth. And what holds them together? It's those hydrogen bonds we talked about, forming all along the backbone of the chain.
So what locks in that final, super important 3D shape? Well, it's everyone we've already met. It's a total team effort. The greasy gang buries itself on the inside. Hydrogen bonds form between the polar pals. The charged amino acids form salt bridges, where a positive hooks up with a negative. And of course, cysteine makes those superglued disulfide bridges. It's the perfect example of how all those different R group personalities have to work together. So why? Why did we go through all of this? The families, the folding, why does it matter? Because in the world of proteins, there is one rule to rule them all. Shape is everything. A protein's specific 3D structure is the only thing that lets it do its specific job. This right here, this is the golden rule of biochemistry. The list of letters, the primary sequence, tells the protein how to fold. And the final folded shape, the structure, determines what job that protein can do. It's a perfect chain of command, from sequence to shape to function. And the jobs these tiny machines do, they're absolutely incredible. One sequence folds into hemoglobin, a machine perfectly shaped to carry oxygen. A different sequence folds into pepsin, an enzyme shaped to chop up food in your stomach. Another sequence folds into an antibody, a weapon shaped to hunt down a specific virus. And get this, the level of precision needed is just staggering. We're talking about just one single mistake, one wrong letter in a chain of hundreds can change the final shape, and that tiny change can break the protein, causing it to not work at all. This is the root cause of so many diseases. It all comes down to getting that sequence exactly right. And that brings us to our final thought for today. We've seen how this beautiful sequence of letters builds a perfect little machine. But what happens if that machine gets broken? If heat or acid or some other kind of stress makes it unfold and lose that precious shape? Think about that, because when a protein loses its shape, it loses its purpose. And now that you know how these machines are built, you're one step closer to understanding why, sometimes, they break.